Okay, if we can have all of our speakers from today come up to the podium, come sit at the panel. So just so y'all know, I have a list of questions from our community for everybody. So this is where I'm going to start because we crowdsource questions as we do every year for those that are online and those that are in the room that might not be comfortable asking those questions or understand the jargon to be able to feel comfortable asking those questions. So I'm going to be representing you, and then we'll open it up to the floor as well. Okay, so I'll start with Jorg since you're in front of me. I'll just go around in a circle. I'll start with you. Well, both of you, you and Shady. So the data that you just showed obviously looks clinically pretty great, and we have never seen that before, so that's really exciting for the community, and I think supports the other ASO trials as well, and that everyone's seeing something relatively similar from a clinical perspective. But the question that was asked was, if there is a known partner and timeline of that potentially still having an opportunity to get to families is the first part of the question. The second part of the question is, if you don't find a partner and the program does not progress, are you willing to commit to share the data with the ABOM and ladder so that we can utilize that data as control arms for other clinical trials?
Thank you for this question. First of all, we appreciate all the efforts and collaboration, the support, the parents and the families who were in this trial. So yes, the data looks promising, and we shared as much as we were able to share in this time frame today. But we will have a publication soon. We are working with many of the PIs on that, and this publication will include like the majority of the data, let's say, and this will help this molecule hopefully to advance to a next stage with a potential partner, but as well it will help the science and the development of the drug development in Angelman syndrome. So all this data will be shared soon, and we hope that will help other pharmaceutical companies, will help the community to proceed forward. Thank you.
Okay, I'll go to Becky because you're next. Let's see. First question for Ionis was, can you please expand upon the specific gains that you have seen in expressive language and communication as that being your primary endpoint? And they also ask communication, cognition, fine motor, gross motor, independent daily skills in the trial, but specific examples as we don't know what those scores on the Bayley and Vineland mean compared to natural history. For my child, even when you put them up and it says they look better, what does that mean? Can you give specific clinical examples?
I might actually turn that over to Dr. Bird, who actually was part of the trial and has some direct experience with that. But I will say that, I mean, we do expect individuals to have individual responses to the treatment. Everybody's unique, and we've talked about that this morning, about how each of these therapies will result in different gains for different types of people. So we do see that if we see an overall increase in expressive communication, that means generally everybody's improving in some way, but specifically how may be different for each individual. But I think I'll turn it over to Dr. Bird, and maybe she can share some direct experience with the trial.
Can we put Dr. Bird on the screen, please? Thank you.
Thank you, Becky, for the opportunity to comment. So we heard things like more vocalizations were being made, more attempts to use communication devices, better understanding and ability to follow commands. So we had a variety of different responses depending also on the age of the participant whose family was responding, but nearly everybody showed some increased interest and increased ability to communicate their needs, wishes, and feelings.
Okay, thank you. Jennifer, you're next. Okay, I have a couple for you, Jennifer, but the one I'm going to start with is knowing your involvement in transformatics and the HSE gene therapy program, which yesterday people were super excited about, but there were no clinical questions asked yesterday. One of them was, do you have any sense, if the HSE goes through, what the timeline could look like for this therapy to get into humans?
That's really hard to say because we have so many non-clinical studies that have to still be conducted to get to a clinical trial application. So I would say it's at a fairly early stage where the IND enabling work has to happen. And once that happens and there's additional regulatory discussions, we'd have a better sense of what the timeline is.
Sarika, you're next. First question for Encoded. When might you expect a clinical trial for your miRNA program? And can you comment on the genotypes and the ages that might be included in that first phase 1/2 if we are able to get there?
So right now, it's too early to say. We are, as I mentioned earlier, we've just recently gotten that newest NHP data, which we're very excited about. We're in the process of critically evaluating our preclinical data package, and that'll help us shape next timelines in addition to regulatory interactions. But as I said, we're excited about the data, so hopefully we'll be able to share on that front in the near future.
Question for Kim, for Ultragenics. I'm going to have to ask you the same question I asked Becky, because that's what's fair. And that's what everyone really wants to know, I'm sorry. Please expand on the specific gains that you have seen. I know you showed some quotes, but it wasn't really so specific in terms of like just excerpts and examples that families are reaching to understand what that means that you guys have seen in communication, cognition, fine gross motor, and independent daily living skills.
It's such an important question because the numbers are just numbers, but it needs to be clinically meaningful. And that's what the regulators expect of us as well. It can't just be a change in a number. It needs to be something that impacts the patient and they improve. So we do have our skills, so our new skills that are acquired over the course of the trial. We had some of those examples. It's things like, increased word approximations in words, being able to understand and follow 1 and even 2-step commands. Fine motor skills getting a little bit better. We've seen improvements in kids being able to attend and focus and be calm enough to sit through dinner with their families and even start to feed themselves independently. So maybe a caregiver isn't having to feed them while they're feeding themselves. So in terms of gross motor, we've seen some improvements in balance coordination. So what does that even mean? It means that they are steadier on their feet, and maybe you as the caregiver aren't having to kind of wrangle and keep a hand nearly on them to keep them from falling, which then means you could potentially go out for a walk with your family, out in public. We've even seen some stories of kids who are able to go on a hike or a walk outside when before they may have some trouble, navigating those steps or cracks or, bumps in the road. They're able to navigate that a little bit better. It's hard to tease those down into discrete buckets. We try to with our different measures, but I think that it's all showing that, this is a potential treatment that is actually, getting at that core root of what's going on with Angelman syndrome.
Dr. Wilson, the first question asked more than once was, is there a world in which you can see a gene therapy and another modality like an ASO working together in order to maximize biodistribution and UBE3A expression?
Yes, there is. In fact, there's, in the SMA world, there's a movement, or there are some patients that receive gene therapy and then Spinraza. Biologically, how it could complement one another, it's sort of hard to know, but more protein produced, probably the better. The only issue is the resistance of payers to pay twice, and that's real. There are some countries that, when there are multiple modalities, you get one shot on goal, but biologically that could make sense.
Great answer. I'll go back to Roche. The next question here was, and it was similar to what a parent in the audience asked, but a little bit different, which is obviously understanding for the deletion genotype, which is majority in oligobad, and the potential for enhancing GABA expression or GABA function for those kiddos, but also understanding when we think about how different deletion and mutation are, which in the grand scheme of things compared to neurotypical are not all that different, which we also understand that UBE3A may have also an impact on GABA, and therefore in your trial you may not actually see much of a benefit of GABA enhancement alone, but you might see a dramatic benefit once you replace UBE3A of GABA enhancement. Because of the impact UBE3A has directly on GABA2. So, and I think for DUP15 we see the same UBE3A versus the GABA genes, which is responsible for most of the phenotype. It may be the UBE3A impacts GABA overexpression. Maybe we would see the same for underexpression. So the question really is if you don't get a signal with alogabad alone, but you might get a really great signal with alogabad in an ESO, are you missing that opportunity, and how do you consider that?
Yeah, I mean, I understand that question. I mean, my colleague Shady elaborated on the complexity of developing two drugs at the same time, both of which are not approved. So I think for the time being, I mean, the study we are conducting right now is a proof of mechanism study, so it's not targeted at efficacy. I mean, efficacy is an exploratory endpoint, but there we will get an answer if, let's say, the physiological signature we see in the EEG is corrected to a substantial degree, which would then suggest it makes sense to continue the development, just to clarify that. Yeah, then if that's the case and one continues and one would run an efficacy trial and does not see efficacy alone, yeah, I think you're right, one would potentially not proceed to a combination trial and could potentially miss this. Yeah, I mean, this is in the nature of the complexity of the things.
I think it goes back to our mission of collaboration in that anyone who gets an ASO approved, hopefully many of you do, maybe we need to really think about how we wouldn't let that die on the vine if it doesn't make it through on its own, but we can think about looking at that as next steps for the deletion population. Collaboration is key. Okay, thank you. Becky, I'm gonna go back to you. The second question here is, what is the status of your CSF UBE3A assay that was reported a few years ago, though we haven't heard about that for a long time, and we're wondering how that played out in your clinical trial?
Good question. No, we didn't report on CSF today. So I think everyone remembers several years ago we did a pre-competitive collaboration with Roche and Biogen and Ionis and embarked on a CSF natural history study where we collected that. And the purpose of that was to be able to develop an assay to measure UBE3A in the CSF. And it was successful. We collected the samples, we were able to develop an assay, and we've shown in the past that we can actually measure UBE3A in the CSF. Having said that, upregulating neuronal UBE3A in the brain with ion 582 in the HALOS trial did not result in any change in CSF UBE3A. And we think that's probably because the production of UBE3A in the CSF is coming from so many different cell types, not just the ones associated with Angelman syndrome. So it's likely that there's still a large amount of UBE3A protein being made from other cell types, and that's why we're not seeing necessarily that when you upregulate in the brain, we're not seeing it in the CSF.
Interesting to understand how the UPD or ICD kiddos, which probably are not haploinsufficient in those other cell types, compare to the mutation and deletion at baseline, because that would answer that question, which would be really interesting. Great. Okay, Jennifer. We talked a little bit on Thursday at ABOM, and then yesterday it was mentioned in the first sessions about MRI in Angelman syndrome and understanding gray matter, white matter changes in the Angelman syndrome brain and how we might be able to consider using that as a biomarker for Angelman syndrome and also for dosing when it comes to brain dosing and understanding CSF volume and brain volume. Can you comment on the value of potentially using MRI as a biomarker in Angelman syndrome?
You want me to answer that? Yeah, at the ABOM,
you're the co-director of the ABOM.
No,
So
I
from
hear
an ABOM
you. I hear you.
perspective-
I'm just joking. At the ABOM, we did see a presentation regarding differences in white matter volume in particular in individuals with Angelman syndrome versus neurotypicals. And we do know from other neurological disorders that MRI is a very powerful biomarker, and there are many groups that can look at volume of brain, so they look at brain volumetric analyses to look at response to treatments. And so because of that, and because it's had such a powerful role in understanding response to treatment with other therapeutics for other neurological diseases, it is something that we're going to invest in as part of the ABOM to understand if there's a potential biomarker there. And we're very much interested in biomarkers from the standpoint that they help underlie some of the more subjective assessments that happen in studies, and MRI, EEG, and blood tests or CSF tests are things that objectively tell you about response. So they're very helpful in interpreting overall effects.
A question for you. Knowing that there was a question in the audience today, and certainly there's been many online, for those individuals that are in the rugonersen trial that are now not going to be able to get that ASO unless there's a partner for a long time, and they'll certainly, by the time you get to clinical trials, be washed out, or one would think they would be washed out by that point. Though I was allowed to announce that the half-life of rugonersen is 7.6 months. Weeks, 7.6 weeks. For anyone that wanted to know, that was a big question. What's the half-life? So we can understand times 5 washout to ensure it's gone. 7.6 weeks, according to Roche and their animal data. So knowing that, would you be willing to include those kids in your miRNA trial? Because by the time you get there, they'll certainly be washed out.
Oh, to me. Yeah, I mean, I think that would definitely be something that we would be considering as part of our clinical development plan. There's certainly a lot of considerations that we'll have to take into account as we think through which patients make the most sense as we shape those clinical trials. And so that will absolutely be a consideration as we think about the patient community and what would be best in order to have a rigorous clinical trial for our candidate.
Thanks. This is for Kim. We know as we think about multi-treatment therapies and single treatment therapies, and we see a pipeline of single treatment therapies coming right behind the ASOs, there is some thought of how we can just make repeat dosing easier for individuals living with Angelman, knowing that a far majority of them require gas anesthesia or general anesthesia and are not able to sit through an LP without sedation or anesthesia. And knowing that those doses are going to be every 2 or 3 or 4 months down the line for maybe the rest of their life. Have you guys considered using devices like catheters or ports as used with Brineura for enzyme replacement therapy, or they're looking at that like the Alcyon device for Spinraza and things like this? Have you guys considered that for GTX-102?
That is a great question. Because we understand it's very exciting. We have lots of opportunities, lots of new exciting science, several clinical trials running, right? Our focus right now is really on our pivotal phase 3 study, because the first step is getting it approved, right? So we want to do our best job to do the most rigorous study we can for our phase 3, get really good, clean data, and convince our regulators that this drug is safe and effective. So with that said, we're always thinking of how we can do better in the future and always looking ahead and trying to understand what are the next steps that we can do while we're hyper-focused on getting this first step done really, really well. So we've got a great team at Ultragenyx. We've got great minds thinking about this, hearing from you as the community that you want us to do our very best, and we're trying. So I think the future looks very bright, but I don't know exactly where we'll end up, but just know we'll keep innovating.
This is for Jim. Do you feel, knowing that with gene replacement therapy, we believe that there is a long durability and duration, but we don't know how long that is, and it may wane over time. And we've talked a lot about redosing and how we could do that with viral delivered therapies. Do you think there's a world in which we would be able to repeat dose, particularly in a low-dose cohort where we know that we are going to potentially need to have multiple dosing in a single treatment therapy, that those individuals will eventually be able to get a higher, potentially more effective dose?
Right. I mean, it's a real challenge for families and for the development of the product. The thing about injecting a vector in the cerebral spinal fluid is there is tolerance to the presence of antibodies against the vector, which is what occurs when you try to re-administer. It also, there are existing antibodies to these vectors just based on natural infections. So all of the products in which you inject the vector into the blood, the Zolgensma, the Duchenne, Sarepta, Roche product, you have to be pre-screened for antibodies, and if you're antibody positive, you're not eligible. The good news is, even if you're antibody positive prior to receiving the vector, it doesn't impact at all on the delivery of the vector. So we know that. We've studied that in non-human primates, studied it in patients because there's no ineligibility as a result of antibody status. The question is, the antibody response that occurred to first treatment, could that impact on re-administering vector in the second time? And definitely if there's a period of time in between, it's going to help. But there are also drugs now that are being evaluated for those trials in which antibody precludes participation to transiently diminish antibodies. And these are approved for other indications. And in those communities where you're either in or out due to antibodies, it is a painful and very compelling problem that there is a lot of work being done. So especially if you're at a low dose, your antibody response will be less. So I would not rule that out.
Okay, I'm going to open the floor. I have a lot more questions here, but I also know the audience is raising their hand even when I told them that it's not their turn yet. So I'm going to let the audience ask a couple questions. There you go. She's been raising her hand for 45 minutes.
I've got two questions, sorry. So the first one's around the GABA receptors, and we saw quite a bit yesterday with the CRISPR and the conversation today. Do we think, so in the general population of epilepsy, about 15% of people have drug-resistant epilepsy, but it's quite common in Angelman syndrome. Do we think that's related to just the loss of UBE3A, or do we think that's related to the GABA receptors as well, or is it a combination of both? And therefore then, from that, do we think any treatment will help with the reduction in seizures and therefore less drug-resistant epilepsy?
Yeah, it's probably a combination of both. So if we look at, from the natural history data, at seizures in mutations and deletions, you see that individuals with deletions have a much earlier onset, and it's also much more complete. So from 1 to about 3 years, it really ramps up, and then about 4 or 5 years, everyone had an onset of epilepsy, while for the mutations, it drags out longer and is also not fully complete. So that clearly gives a hint that these genes, besides UBE3A, have some contribution. And from there, it's speculation that, or a hypothesis, how much the GABA genes are relevant for this. There are some other genes, HERG2, CIFIP, in that area that could also play a role. But yeah, it's plausible that the GABA genes do their contribution. And so it needs to be tested. I think there is a rationale for that.
Thank you. And then one other question, it's to do with the ASO, so it's general, although I think we've answered it for was it Roche? The half-life is 7.8 weeks, I think you said?
7.6 weeks.
7.6 weeks.
So that's basically extrapolating from non-human primate data, scaling it.
Is that the same with all the other ASOs? Are they sort of a similar half-life? And if so, why are we dosing everyone at 12 weeks? All of the studies seem to dose at 12 weeks rather than at the half-life point.
So no, we definitely can't assume that all drugs are the same. They can have different half-lives, different potency. So while the mechanism of action is generally the same, but they are also, targeting different, let's say, pieces of the UBE3A antisense. I think you cannot just consider they are all the same. So basically, the other companies have to share their half-lives. In terms of, so I can only speak for Roche for that reason. Why did we choose 4 months and not more frequent, coming closer to the half-life, which I think really is supported also by the EEG. So the reason being that there were some preclinical studies that led to a decision in the US to have that as a prerequisite to not dose it more frequent. I think there is an opportunity to revisit this with all the data that is out there now for our molecule. But for the other molecules, I don't know if you want to comment on that, Becky.
I can just say that the half-life only tells us about how the drug moves through blood and CSF. There's also another component, which is the pharmacodynamic effect. You might hear PD a lot, and that's about how it interacts with your body and changes more clinical measures. And so we do find that with ASOs in general, the PD tends to be a lot longer. Those effects last a lot longer than they do in the blood or the CSF. So that goes into our decision also about how frequently to dose or infrequently is based on how long we're seeing the clinical effects.
Sorry. So I just want to briefly comment on that. So thanks for bringing that up. I think that's really important. I just wanted to stress that what the EEG data that I showed you suggests is that this lag, how long does the PD persist basically after the drug is gone, is not so long as one may have thought. It seems to be a relatively dynamic effect. For sure it will be delayed, but I was really surprised seeing the data that it is relatively dynamic. Just to add that.
One question for Neuron. Me, Thomas. So Neuron seemed to suggest, your CEO, that they're going to look after 2 indications and may look at selling one. And I was just wondering if there are any plans for Neuron for the Angelman syndrome indication to move to phase 3, or what are your plans there?
Great. Thanks for the question. Currently, we are actively reviewing our data and assessing a path forward. So no decisions have been made, and we're still very much evaluating the data and looking for the best path forward.
Okay, thank you.
I have a question in the back.
Yeah, all the way in the back here in the middle. Hi, my name is Sam. I'm with the Brazilian community. We've been coming to the summits for about 9 years now, and we've witnessed each step of the way, from the initial research and animal testing, and then the anticipation of FDA approval, and then now the clinical trials. So as phase 3 trials begin with another 3 years to go, many of us are holding on to the hope that our children might participate. However, due to the strict criteria related to age, genotype, or location, many kids are unable to join. So in our community in Brazil, for example, we have kids that have over 100 seizures a day, and so even the smallest of improvements in quality of life would mean the world to these families. So my question is, given the success of phase 1 and phase 2 trials with both Ionis and Ultragenyx, is there any possibility for implementing compassionate use or early access programs? Because maybe it would be a lifeline to these families. Thank you.
I'm happy to start with that. Yeah, so, and every company is going to have a different policy towards expanded access. I mean, our first and foremost priority is testing the safety and efficacy in a controlled trial, and that's our fastest path to get a potential drug approved so everyone can access it. And depending on the regulatory body that's reviewing it, whether it's FDA in the US or Anvisa in South America, they require you to have demonstrated safety and efficacy, at least in clinical trials, before you open it up to the broader population. And so as we're still in clinical trials, that data hasn't been established to the level that we could open it up to different age groups and genotypes beyond what we've already tested. So for Ionis, our goal is to get through the pivotal phase 3. That's our most efficient path to get it approved and then open up broadly for the general population.
Hi, yeah. So my question is both for Halos and the GTX together, really. Just about the results are quite obviously significantly amazing so far. But I was wondering about, is this across the board with all children? Are they all showing signs of improvement, or does it depend on severity? Because I think that's the one thing I didn't really see today around, obviously all our children are very different, and some struggle more with their motor side than other children. So has that made a difference on how children have responded throughout?
Yeah, I'm happy to take that. So yes. So I think what is really exciting to me is, especially my former hat wearing as a clinician, being in clinic with families like you, is we look at the data as a group, and that's there's always a process of how you're analyzing these data. So you look at it as a group, but the power of our multi-domain responder index is really just that. We know that Angelman is not one thing. It's multiple symptoms, and some kids may have more motor disability, other kids have more sleep disability or sleep disruptions. And so when we look at our multi-domain responder index, that's exactly what we're doing. We're taking that data that we had just looked at in aggregate for the whole group, and then looking at it on the individual level to see who improves in different ways, and who responds in different ways to these to this drug. And I think we see that we are getting kind of a really interesting pattern of response that shows us that it really is changing different aspects for different patients. And so I think that's really where we get really excited looking at our data.
A follow up on that, and then you guys, but let me, because there's two questions for you guys on that, which is the same concept, which is age. And there's a broad range of ages, 4 to 17 and 2 to 50, and you guys were a little younger, 2 to 12. But would you say, and we've heard you mention this before in the past publicly, that you didn't appreciate a difference in skill gain with age for the Ultragenyx trial, that they all gain skills regardless of their age and of similar quantity and similar types, but different per patient. Did you see the same, Becky, in terms of age? And then for Becky and Roche in terms of genotype, because they only had deletion, but did mutation or deletion seem to do better or worse or similarly? And then did age seem to have an impact on overall delta of change?
Yeah, good question. Yeah, we did. We looked at a pretty broad spectrum in the HALOS trial, and the sample sizes are small, especially when you start cutting them into really different groupings. Having said that, I'll take the age question first, from 2 to 50. And what we saw was that no matter where you started, and no matter what your age was, the majority of subjects in the trial did show improvements across all of the functional domains that Dr. Bird showed earlier. So it didn't matter how old you were or where you started. The majority of participants did show improvements across the board. In terms of genotype between deletion and mutation, of course we had a lot more individuals with deletion than the mutation genotype, but when we look at that again, we could see that their baseline level of functioning was different. As you would expect, the deletion had more severe impairments than the mutation. But again, having said that, they both responded fairly equally in terms of their gains during the trial across the board. So far in our hands, in a small sample in an open label trial, age and mutation has not made a significant difference in response to ION 582.
Mice are not humans. Roche, you want to answer that for Roche as well? I'll give that to you 1 second. I'm just going to let you guys.
Yeah, I mean, so we had a narrow age range, so 1 to 12, so there are some differences. We had 7 of the 61 individuals being mutations. So also coming to these, so we'll provide like complete analysis of all of this in the manuscript that we are currently writing up. So similarly to what Becky said, we don't find really, given to consider the limited sample of 7, but we don't find clear differences in terms of genotype. So we see both for EEG and for the clinical scales, similar magnitude. For the ages, for the clinical scales, it is also comparable. For the EEG, funnily, or not funnily, but surprisingly, I would say, the effect is weaker for the younger ones, significantly. We don't exactly know what that means. It could, I think it's an interesting observation. It could point to that, so let me just speculate for a second. So it could mean that the drug is differently metabolized in younger ones. It could mean that the organism needs more UB3A to have similar changes. But to be honest, we also don't exactly know if this a certain change in an EEG means the same for younger compared to an older. But just to point that out, so we see a difference there.
Right, last question, because they're standing waving their hand. So go ahead, Isabel.
Hi, good afternoon. I wanted to follow up with from the dosing comments earlier. We've been hearing a little bit about frequency, a little bit with Roche, and then we've heard within the community as well how ASOs may start to wear off in some kids at 1 to 2 months post-dose. We've heard Ultragenyx is willing to explore more frequent dosing at every 2 months, and I'm curious if Ionis is also considering increasing their dosing frequency to help with kids that may be experiencing withdrawals.
Sure, good question. So in the HALOS trial, we did study different dosing frequencies in the beginning. So in the very beginning, in the MAD part 1 that we showed today, we had a 1 month dose interval and then a 2 month dose interval. And then in the long-term extension, everybody went to quarterly dosing. So limited amount of data to drive some of those decisions. But having said that, and we have heard that from the community as well, is that there are some cases where there's a perceived wearing off effect, and does that mean you need more dosing frequency? And so we continue to look at that in the HALOS trial in the long-term extension. We do have people on quarterly dosing now. I think we need more long-term data to understand that and how the drug responds over the long term. It may just be that those first 6 months they didn't have enough time on the drug to be able to see how it changes over time, and how drug may build up in the body and last longer with more dosing. So I think those are things we're still looking at in the HALOS trial, and if we get data to tell us that we need to look at different dosing intervals, we'll certainly do that.
Just adding to that question, can a primary investigator of the trial maybe comment on the withdrawals and the implications on the child?
I know EBK is in the room. Oh, EBK is coming up. We'll give her an opportunity. There we go. And then Lynn, look at you as well.
So I think in general in all the trials we've been a little bit surprised that the ASOs don't last as long as maybe in some other conditions, and that probably has nothing to do with the ASOs themselves, but more to do with the UBE3A and the fact that it may be just a protein that goes up and down more easily, and the things it regulates may be more acute things and less chronic kinds of things, or maybe just to do with the kinetics of the protein. I think we don't understand it. But I would say, having dosed 40 patients at my site, it is a phenomenon. I mean, we thought the Roche molecule would last longer than it does, and definitely with the 6-month dosing, we clinically saw the wearing off and kept telling Roche before the EEG data told us that that was probably the case. And we know that in the other studies we have seen wearing off. With the Ultragenyx study, it's been more, in genetics, it's been more complicated because of all the dose changes that have had to go along with the response to the radiculopathy, so we haven't always been sure that it's just not that the patients are underdosed. But there has been a response in terms of allowing us in the long-term extension to explore both 2 and 3 month dosing with different doses so that we actually get some data on what's working better in terms of the dosing. I think our Ionis patients, we have about, I would say, 50 to 60% who have a definite phenomenon at the end where and they will tell us this week is when it happened. Like, we could see it. School saw it, even though school may not even know the patient is in a trial, and we see sleep and behavior, which are the most friable symptoms, obviously, tailing off. So I believe this is a real phenomenon. We've actually tried to think about ways of tracking it during the dosing interval, like having people rate 1 to 10, how was sleep and behavior, and then watching it. Because it really is, for some of the patients who have a more dramatic withdrawal, quote withdrawal, we don't know if it's really a withdrawal. There's a roller coaster effect for them, and they really experience deterioration in quality of life in the last couple of weeks before they come in from dosing. And I know we don't really have data to look at that because we haven't been assessing them 2 months into dosing and then again 3 months into dosing. But I think we really need to work this out because we wouldn't want to have a drug approved with a dosing frequency that was going to result in some patients sort of doing this over time. We would want to have options for insurance to be able to approve whatever dosing regimen fits the patient best, just like what we do with all drugs where there are different frequencies and doses, amount of doses that we use. So maybe Lynn has some commentary on that also, but or anybody else who's here.
Then this is the last comment because I'm being kicked off the stage. Like, I kicked you all off the stage, now they're kicking me off. So Lynn, do you want to do last comment? And this is a really important question, so I'm grateful to all of you because it's a conversation in the community. We hear parents saying this, and I'm glad we're bringing it up to industry who may not be hearing it because they're not talking to the patients directly. So this is a really important question.
Well, I do think industry has heard it because the investigators have raise these issues. But I think it's important to note that in the way most of these trials were designed, we were measuring patient performance immediately before the next dose, so at the trough. So it's pretty incredible how much improvement we saw when arguably the drug was waning in effect, at least the effects we could easily hear about from families like sleep and behavior. And I'm not speaking purely for the Ionis trial here because all the patients just kind of come together in my mind. But I think that we had many families telling us that sleep deteriorated, behavior deteriorated, but I specifically inquired of families who had had long dosing intervals, do you think anything that got learned then got forgotten? And families were pretty clear that any skills that were attained were retained, even when sleep and behavior started to fall away. So I'm hopeful that yes, with more data, we are going to find, the optimal dosing interval, which may need to be very individualized. But the signs are there, in my opinion, that even if we don't have the perfect dosing interval for the trials in phase 3, it's good enough to be able to show an effect if what we believe we saw from phase 1-2 is real.
Well, thank you for all these comments. This is patient-focused drug development. What is important to these families? Losing sleep and behavior is awful when you gain it. So even if they don't lose skills, if they lose sleep and they have more anxiety and become more hyperactive, that's horrible for these families. So thank you, Lynn, for saying that. I think that these families deserve for everyone to have heard that. So thank you all. It's been a really, really long 3 days. We have a really exciting night ahead of us. Tonight is where actually the magic happens. We celebrate everything you've done for your children, everything your children have done. And this is where we can raise the money in order to be able to support all of the research that you heard over the last 2 days. Thank you all. It's been a really great couple of days, and we really appreciate all your comments.